Okay, so let's review. Here's the picture of the action potential. Your diagram will probably include this graphical representation of the action potential, surrounded by maybe images of what the ion channels are doing, or at least arrows to each portion of the action potential, explaining what's going on during each of these phases. For example, here we have local potentials that are summating in order to reach threshold. Once the threshold is reached, sodium gates fling open. They work very quickly. Sodium rushes out of the cell, which increases the membrane potential, depolarize it. The depolarization causes more sodium gates to open. It's a positive feedback effect until we reach the plus 35 membrane potential. At this point, the potassium gates begin to open because they function much more slowly. Sodium gates have closed, and so potassium is running into the cell very fast, returning the potential to below resting membrane because they're so slow they forget to close on time. Eventually, we'll see the sodium-potassium pumps bring us back up to resting membrane potential. So we have depolarization, repolarization, hyperpolarization happening. During an action potential and for a few milliseconds after, it's really difficult or impossible to stimulate that region of the neuron to fire again. This phenomenon is called the refractory period. This is a period in which there's resistance to stimulation, and it happens for a couple of different reasons. There's an absolute refractory period and then a relative refractory period. The absolute refractory period means that no stimulus of any strength will trigger an action potential. This has to do with the sodium inactivation gates being closed. So that goes back to the example shown in the animation. When the inactivation gates are closed, even if the activation gates opened, we couldn't possibly elicit a response. The relative refractory period means that only an especially strong stimulus can trigger another action potential. The potassium gates are still open during the relative refractory period, and thus potassium is still moving out of the cell, and we're hyperpolarizing, as in becoming more polarized. Thus, it takes a greater stimulus to raise us up to the threshold value. The refractory period is only occurring in a small patch of the neuron's membrane at one time. The other parts of the neuron can be stimulated, while this part is in refractory period. What this allows is for signal propagation to move in one direction. Let's look at that. When we look at conduction in unmyelinated fibers, the nerve signal needs to travel to the end of the axon. The unmyelinated fiber has voltage-regulated ion gates along its entire length, and the action potential is sent from the trigger zone, causes sodium to enter the axon and diffuse into adjacent regions beneath the membrane. The depolarization excites voltage-gated channels immediately distal to the action potential, and the sodium and potassium gates open and close, producing a new action potential in the membrane. By repetition, the membrane distal to that is excited. A chain reaction continues to the end of the axon. Let's view another animation.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
In myelinated fibers, we have myelin from the Schwann cells or the oligodendrocytes. In these regions, we will see far less sodium and potassium channels. It's difficult for sodium and potassium to cross the membranes at this point. There are fewer than 25 of them per micrometer in the myelin-covered regions, and they're up to 12,000 at the nodes of Ranvier. So fast sodium diffusion occurs between nodes. Some current moves under the internode and again excites the channels that are gated in the nodes of Ranvier. In this fashion, the nerve signal seems to jump from one node to the next. We call this saltatory conduction. So the basic story of saltatory conduction is that the action potential can only be signaled in the nodes of Ranvier where our voltage-gated channels are. In the internodes, there's way less channels, so there's way less transport of sodium and potassium, but enough to excite the gated channels at the next node. Here's another image of saltatory conduction. It moves in the same way down the axon as regular conduction, but it jumps from node to node to node. Conduction is much faster in myelinated axons than it is in unmyelinated axons because of this jumping distances along the neuron.